Oh, I like the look of this boulder off to the right here.
Oh,
This
yeah.
feels like a good one.
Sizable.
Very basalty. Oh, it's got a whole ridge action.
Ooh, yeah, cool Argus too.
I'm heading kind of east more than north, but
That's all right.
Little channel
Rock.
here.
We got rock.
We got fish too.
You want me to step towards you?
how far are we gonna go this way? I guess forever.
We should,
Sure.
yeah, we want to,
Yeah, we can do a 045 or something.
yeah.
Are we done exploring the southwest portion?
Southwest, yeah, I think. So now we're like kind of on the northwest
Ooh.
portion, right?
So I want to go 4 or 5.
Look at there's a sponge
Yeah.
tucked in that crack there.
Yeah.
Oh wow.
Glass
And interesting looking crinoid question mark?
Yeah, zoom in, please, Ed.
What is that?
That white spot?
With the things, is that a
Mussel?
that's a no, it's a squatty.
Urchin?
Squat urchin.
Urchin?
Urchin.
Oh man.
Urchin, wow.
Wow.
Can we push it any farther?
Sure. Oh, Sam, what have you done?
I'm gonna go 4, 5, 3.
Roger.
Bridge now.
Nice. Yeah, that's one of those. Oh, and you see that bubble on top?
1 step,
Eggs?
30 meters, 045.
Nope. I'm happy to report
Thank
that
you.
that bubble on top is an anal
you.
sac.
Oh,
And
nice.
those pellets you see are fecal pellets.
Huh.
Cool.
Why do you think it just holds it?
Oh.
Oh, it's
Oh.
not.
It doesn't
It's-
hold it.
It's letting,
Gotta go, you gotta go.
it's letting go.
Yep.
Right on. Okay, let's let him have some privacy.
It's a little floaty thing.
That floaty thing was very stationary.
Yeah.
So I think that genus is Aspidodiadema.
Yeah.
Yeah, sorry.
Aren't diademas the long-spined sea urchins that we find in shallow water?
Diadema? Yeah. Yep. So
Venomous ones.
that must be, let's go find out what diadema means.
Commonly known as long-spined sea urchin.
Diadema is a Latin word. Oh. Yeah. Meaning tiara or
Yeah,
crown.
That's from Harry Potter.
I don't know if it's from there.
No, it's not from there.
Yeah, in Spanish, if you say tiara, it's like diadema. So
Nice.
it's more or less the same thing.
That's cool.
So you're stepping northeast.
Yes.
So that'll allow Trevor to kind of go up and over this
Yep.
feature. Yeah.
What's right in front of us? Oh, just a rock with
With
Oh,
yellow.
what's that
mineral.
yellow?
Oh, this
Huh.
is new.
There's
Some
something weird on the.
weathered.
That looks like a rock that wants to be sampled.
Yeah, it does.
The one
it's-
up top of the left.
Usually
Yeah.
they don't want altered samples because
Oh.
you can't. Doesn't have what they.
You mean colored?
Yeah, it's been altered. Yeah, so chemically altered by.
Something. You don't think it came from this vein here?
I think it's altered. I don't think it's. I don't think it's
This flow.
like a different
Zoom in, please, Ed.
type of rock or anything. It's basalt that's been altered.
This is the one you're looking at?
Basaltered.
Basaltered.
Basalted rock, yeah.
Oh, but it's got a nice little coral on it.
Hmm.
Indeed.
Like in order to do argon dating, they would need non-altered bits, so that you would get a piece from inside that. But
Hmm.
There's a brittle star at the bottom there. Oop.
There was a brittle star at the bottom. Oh, yeah.
Okay. No interest in sampling this?
No.
Okay. Absolutely not. What do we think the yellow is?
Right. Let's take another look at it. Off to the right. That
Sure.
little strange. Didn't see that on the seamount.
Mm-mm. Lots of those round sponges there too in the cracks.
Okay, Ed, go
I
ahead.
don't know. It's like oxidized.
yeah.
Oh, tunicate.
Oh.
Oh, wow.
Nice. Well, that one's even more see-through.
Is he gonna say ah?
Someone yawn.
Bridge nav. One more step. Thank you.
Okay, see what we want?
Yep.
Come on.
If there's a loose, like, non-altered piece or something, that'd be something to grab.
It's a cool ridge. You can see the
Yeah,
Argus
it's
view
really
there.
cool.
How far more? How much left on this step?
We finished one, and
Okay.
I called in another.
Another to the northeast.
Yeah, I did. I can stop it
Maybe.
and readjust.
Yeah. Maybe we want to stop and allow them to move east.
Okay. Bridge nav. Let's hold position here. And then you'll go follow that ridge, I guess, to the right.
Sure thing.
Anything on sonar?
Not much, no. There may be that little Argus shadow, kind of. Oh, that's south of us. Never mind.
That's the future,
That's
I
the
think.
thing.
Yeah. But something's wrong with Argus's position.
I'm not happy with it, no.
No, because we're facing 50 and looking at you.
Yeah. That's better.
That's yeah. Keep that.
Okay. Let's look at this thing in Herc view, or in Argus view. Coming into view now.
Holothurian on, or no, that's floaty. Sorry, I was looking at the wrong
this,
thing.
looking at the seam here.
I was,
It's a really neat looking seam.
hey, look at that. That's what you want.
Okay. We moving east now?
I don't think you ordered that.
I
move
held
yet.
position
Yeah.
right here so we could
Yeah,
figure
we'll
out
check
which way
this
we
out.
want to go.
So
Okay.
I don't think that, Herc and Argus haven't been over the center of the bright target yet.
No, we're just circumnavigating it.
Yeah.
So where do you want me to go?
So, yeah, I think kind of east-southeast.
East, southeast.
Just a little.
Roger.
10 or 20 meters or so.
Sounds
Yeah,
good.
I'll hold here until we figure out
Yeah, I think
what,
you got
what it
enough
looks
tether.
like. Yeah.
Might as well keep the seam in view for now.
There's the ocean drain.
It's kind of cool. It looks like
Yeah.
a little coastline.
Yeah, it does,
And
doesn't it?
Hercules
Is what?
Please
I
say
couldn't-
in the water.
yeah.
Yeah, it's a cool seam. Seems to keep going though. I'm gonna keep going east-southeast now.
I think that this flow is causing this big bright spot. I think that's what's under there.
Oh yeah.
Like that boulder wasn't just, that was something in the area, but I think that this,
But this is all just barely covered?
yeah.
I gotcha.
Babby Argus.
Babby.
Future Gabby.
The future winch makes me so happy.
Yeah.
I was surprised when we measured the difference in those, how little it was. I expected it to
Yeah.
be a couple seconds.
I expected 2, but you said it was like 3/4?
It's 11/30 to 22/30. It's variable.
So half.
Yeah.
So maybe we go, yeah. Where do you want to go from here, Emil?
Let's see where we are here.
Yeah, you can see we kind of done a circle around this.
Yeah.
Kind of.
Yeah, a little bit more southeast, and then we'll head on up to the,
gonna
just to like 10,
step
20
it.
meters, yeah. And then we'll head up to
Step by.
Step by.
that web 1929.
Bridge now. 1 step 30 meters, bearing 130. Thank you.
I think I'm going to try a different nap, eat, gym technique today. Shorter nap.
Yeah.
Haven't really found the groove yet.
Doesn't look like much out there.
Not much this way, no.
Close the loop and drive through it. That'll cover it.
Tie a bowl in.
Tie bowling.
Is that what we're doing?
Still, ship's still moving a bit, so.
What do you think, Emil?
What's that? So yeah, you're waiting. We have a move to the southeast out in.
Yes. Just starting, yeah.
Yeah, as soon as we're finished that, let's head up to that web 1929 mark. It's kind of a topographic high there.
This one over here,
Yeah.
yeah.
Be a fairly steep ascent.
Yeah. I'm sure they'll get some good stuff in there.
Yeah, I think it looks like maybe the next 100 meters be sandy if the backscatter's right, and then we start getting into some good stuff again.
So line of sight, we got about 535 meters, but what we actually did was 350 plus 270.
Look at this.
So
What
six,
is
just
that?
over
Is that
6.
line?
Huh.
That's
Oh,
ooh,
What we got
That's
there?
interesting.
man-made.
yeah,
Man-made?
Man-made.
It looks like it's all on the bottom.
yeah. I don't see anything in
Yeah.
Argus. What kind of line is it?
Interesting
Let's zoom in
the
on
way
it.
it's landed
Yeah, I'll
like
get
that.
closer first.
Yeah,
Yep.
it really landed all in a pile like that, huh?
Yeah, that's
It's like they
wild.
just dropped it off.
Yeah.
It didn't get caught. They just threw it,
It
like
looks
dropped it
cut.
off. Yeah.
Might have a
What's-
loop at the one end. There's
It's nice to be able to see both ends. Can confirm.
Yeah.
Yeah.
what's that in the center? An iPhone?
I think so, yeah. Okay, zoom in, please.
Yeah, coming in slow.
That's a twisted cable.
Yeah,
That's cable,
Heavy,
yeah,
that's
it's
why.
cable.
like steel.
Oh,
Doesn't-
tuna kit.
Hmm.
Doesn't look that old.
No, it doesn't.
There's the loop.
Holding there.
Wonder how it got here.
Is that steel?
I don't see any orange.
Yeah, I don't think it's line. I think it's steel.
Yeah.
It
it
looks
is.
oxidized. in certain
Yeah.
parts.
It does.
Oh yeah, there's a little, but not a
Alright.
ton. Not like it's been here for very, very long.
That's what would explain why it fell altogether.
Hey, Trevor, can you
Oh, we have some.
back down a little bit and back up a little bit and look at that ledge?
Come wide, please.
I think there's another loop up there too.
Yeah.
Just given how remote we are, it's just amazing to me that someone else has been out here.
Yeah. That's the first trash we've seen.
Yeah.
Yeah. And it's super weird how it dropped like that.
Yeah.
That's
Yeah.
not usually what we see.
It must have been there for at least a little while because I could see some little invertebrates colonizing it.
Yeah. A couple barnacles, it looked like.
I'm not really surprised to see that it fell like that because it's so heavy.
Yeah.
Huh.
I wouldn't see
true.
that with line with
Yeah.
yeah, but with steel cable, I'm not too
Just
surprised.
follow this along to the right.
Roger.
Looked like a nice cable though.
Yeah.
I wonder if the other side was also looped or if it had snapped.
It looked like it was.
It looked like looped, yeah.
Yeah.
Huh. So it was intentional.
Is that a shrimp under there?
Mushroom coral. Just slam a piece of this off.
That's pretty.
Okay, I can't stay too much longer.
No.
Very slabular though.
Ship move's complete though. I'm sure Argus has a bit of sway, but
Okay,
Yeah.
Emil, do you want to keep moving or do you want to not?
Let's just keep sliding along this if you can. And
Sure.
then when we run out, we'll move on. Yeah, just follow it on up. It's something to the right here. Yep, this is what was given the backscatter return.
Yeah, that makes sense.
mhm.
And that I'll wag over, but I think that's the end of it.
Okay,
Ooh, what about these rocks?
What about them?
Those are rocks.
Certainly are.
Yeah, I don't know. We haven't collected any samples yet.
yeah. Are you able to look at what's behind you? Or do we need a ship move for that?
I can go back about another 4 meters kind of
Yeah.
thing.
All right.
And that's about the limit-ish. I can look to the right here.
Yeah. No.
That's kind of where we were, looking back
Yeah.
towards where we were.
Yeah.
So
Yeah.
All right, we close the loop.
pretty much.
Loop closed.
Okay. Yeah, let's move on.
Okay.
Okay, heading towards that topographic high?
Yep.
Okay. Bridge nav. .30-meter step, bearing 060. Yes.
Yeah, so for those asking at home, we're expecting this dive to be at least 20 hours. So by the end of our watch, we will still have at least 16 more hours to go. So stay tuned.
Subject to change.
Yeah, it can
Yep.
always change.
Looks like watch change is happening.
Hey. Okay. I haven't seen a whole lot of hard substrate. Just
Watch change it video.
Oops.
Yeah, so watch change is about to happen for all the positions here on the control van, so just stay tuned.
Good afternoon, everybody.
Cool, cool, cool. Yeah, good afternoon.
Hey,
Good day.
Sam. Hey, everyone.
Good day, sir. Roger.
Is Nick up here?
Yep, next year. Looks like we have everyone in the front row.
Yeah, I'm not sure if Dwight is planning on
This is Nick.
coming up. He might be working on some stuff.
Smart streams.
All right, looks like we've got the crew here then.
Okay, so we are doing 060. Should we move like, keep going steps until we see something on Argus target, Argus meso? Yeah. Bridge nav, step 30 meters at 60 degrees, please. Thank you.
Yeah, we're just kind of keep chasing either backscatter targets or sonar targets and kind of loosely follow the waypoints, but not too closely.
What was the bearing of your last move?
060.
8. Looks like it was pretty quiet this morning.
Yeah, I think a lot of sand and a lot of rocks.
Yep. Few corals.
Only 2 corals?
A few.
A few. I
Few.
understood 2. I was like, whoa.
More than 2.
More than 2. Okay.
At least 7.
At least 7.
At least 7.
Accurate,
Which
but not
is-
precise.
Which is more than what Google estimated, actually, from what I remember.
Could we do a quick zoom on that fish?
Ooh.
Please? Oh, maybe not.
Gonna say yes.
He heard me. I have
I
to
know,
be
I was
quiet.
going to say,
One
sometimes
more
it feels like
try.
they can hear us.
I'm sure they can hear the vehicle.
Yes.
Go ahead, video, zoom in there.
Yeah, I think it's one of my snake things.
I think you're right, Albert.
You
Al-
It is a snake
Alberta's
fish.
Snakefish.
Just to be clear, audience, that's not its real name, just how I refer to this fish.
Albert, you should write a book with all of your names.
Albert's nomenclature of marine life.
Yeah, no, I don't feel like writing so much. I just did my thesis less than a year ago, so I'm not writing a single phrase in some time.
That's fine, thanks.
Full wide.
Coming up.
Is the scientist who is interested in hiding the fish connecting, or will he recover the images later?
I think he's going to look at the images later.
Okay, it's a pity. Otherwise, we would actually know the fish name probably.
Yeah, I haven't seen him on. It's Ken Sulak, right?
Yeah.
Someone told me that.
Yeah, that's right.
I mean, from what I understand, we just don't know the fish very well in this region, so it might
So
not
we
be
might-
as simple as just looking at a guide or something. It might be.
Okay, so we might not even know the name for like several, like, yeah.
I don't think we know if we know what's here or not.
Okay. Bridge nav. Step again, please.
Yeah, and we're just keep zipping across the sand, but any like fish we come across, we can just do a quick zoom on.
Yeah. Zoom in there, video.
Are we heading towards a waypoint or towards?
Yeah.
Well, this new waypoint, Web 1929.
Oh, okay.
We're kind of just chasing backscatter targets more than
Yeah.
following the actual dive track or sonar targets.
Yeah, I'd say we should go to sonar targets.
Yeah.
It seems to make more
Yeah,
sense.
but there are none so far in the sonar.
Yeah, so I think they were really interested in getting towards this really bright spot
Yep.
up here, which is close to waypoint 2 anyway.
Yep. Looks good.
All right, full wide.
Coming up.
What do we have for samples?
Nothing.
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 I think we could break that record, maybe.
Yes. I thought they tried to get a rock, no?
No.
We're on a mission.
And there's Xenophylophore.
Xenophylophore.
So Alexis was saying they are really hard to collect because they just fall apart really easily.
Like that acorn worm.
Yeah, or maybe worse. So I think they actually had started push coring them.
That makes sense.
Yeah.
We'll bring Argus around there,
And
Gabby.
Sam,
Yeah,
are those
sure.
corals that you're talking about?
No, those are giant forams.
Oh, cool.
So they're really cool. They're actually, I think these, all of them, are one giant cell with multiple nucleuses throughout it. So it's not really a multicellular
Good to go, Mike?
organism,
Video, yep.
even though it's so big.
Wow, that's really cool.
That was that cluster thing on the
Yeah, like the
sand color.
brain looking.
Yeah.
Yeah, forams are super cool.
Yeah, so it's really like very evolutionarily simple, but cool species.
Can I get you a DBL reset?
Yeah, go ahead.
And they're kind of like everywhere.
They're everywhere,
Like-
and they're really like hot in science right now. Like people are really excited about them, and they found them at methane seeps, incorporating methane-derived carbon.
Really?
Yeah, a paper came out like last month. or
Directly or how?
something. Yeah, yeah.
Coming up.
I mean, they
Mike,
can't
Can I
be
take bubble and make it utility?
I mean, if the carbon
Yeah,
that
sure.
they're ingesting is
right. But is it like through? Are they eating something that's eating methane, or are they actually converting methane into?
likely, it would be that they're eating
Yeah,
like bacteria, like
Yeah.
methane-eating bacteria.
that's cool. Those are. Always, I feel like. I mean,
Yeah.
it's not my field,
I'm
but
happy with gauges.
there's been even some like stony deep sea corals where they found like that methane signature very
Yeah.
faintly, and it's
Very faint.
obviously they're not like consuming methane, or they don't have any kind of, as far as we know, symbiotic bacteria that are able
Yeah.
to process methane. So it might just be a little bit of trophic.
Yeah, I
interaction
do
there.
some work on the trophic webs at seeps, and it's super complicated because you have so much going on. You have all this particulates that are floating down. It's so mixed that it's hard to like get that clear signal
Yeah.
sometimes. So you have to use some like kind of weird methods.
Zoom in.
I'm assuming the methane would show up pretty strongly though if it was only like one trophic level away.
It
No?
only shows up strongly if you look at specific fatty acids.
Oh.
Yeah, because there's like certain fatty acids that like we can't produce ourselves. We have to get from our diet. And so if you look at those, there's certain ones that only bacteria make. So then you can really find that signal there. But there's a lot of selective pressure in how
Okay, full
we
wide.
incorporate
Coming up.
food. Like when you eat a burger versus when you eat a salad, your body puts that energy into different reserves a lot of the times. So when you like typically look at the tissue of an animal, you are seeing what they're preferentially incorporating into their tissue, which bacterial methane isn't super nutrient rich generally, so
Sure.
that tends to not be in their tissue as much.
I think if you looked at my fatty acid, you would not find a lot coming from salad.
Tater tots.
Yeah, Zoom.
Tater tots and burgers.
You are what you eat.
That's a helpful analogy, Sarah. I was thinking about that.
All right, full wide.
What might they be consuming that would transfer that methane?
So around seeps, you have in the sediment and in the water column, you have bacteria that are consuming the methane, and they are super depleted in their carbon isotope, which is a signature for methane, and that's their predominant source of energy, and they can eat those just like they eat the particulates in the water or the detritus on the sediment floor. Basically, it just so happens that they're methane-eating bacteria, and that's where they would mainly get that signal from, as well as some other bacteria and stuff around there. But then you can kind of track that up, because smaller critters can then eat that, like copepods could be eating that, which then they could eat the copepod, and you can just track it up that way.
Gotcha. Huh.
Pull back a little bit quicker than that when you do it, Nick.
Okay.
Please. Not like terrible speed, but something in between.
So at the beginning of your thing, you said there's a cell that is huge. With multiple
nucleases?
Yeah.
Huh.
They can grow into like tree-like shapes too. There's like
a single cell.
Yeah, like
But
arborescent
you can see it like on naked eye?
Yeah.
Yeah,
one single cell.
we're pointing out the next one. They're like fist size that we've been seeing. Or
A fist size single cell.
Yeah, we just passed over one a second ago, actually.
What are they called?
Xenophilophores?
Xenophilophores.
Oh my god. Like, I don't remember my high school very well, but
zoom in there.
Like, I always assume cells were small.
Usually that's true. Yeah, these are rare.
And you guys said you can find them almost everywhere, so they're at varying depths and temperatures and...
Yeah. The more that people are looking at them, the more places that they find them.
Cool.
And there's a ton of diversity. There's tons of different types. And these are the same, if anyone's familiar with those classic pictures of the microscopic images of those kind of like kernel-shaped cells, that's a foram. The hard skeleton, kind of kernel shape, you have to look through a super powered microscope to see it, and they range from that to these ones that are fist size that we can see.
What is a super powered microscope?
It's a kind of a technical science term.
Yeah. You haven't worked with one of those, Gabby?
Not as yet, but I want to.
Sit down in front of a microscope. This one's not super powered. There's a lot of like electron scanning, microscopy.
Okay.
Stuff, yeah.
Anything unexpected yesterday night with the...
I understand what this guy's asking.
With the sampling when you
We
recovered the baby?
found a baby squat lobster in one of the corals, and one of the other corals looked like it was getting ready to release eggs.
Oh, wow.
Oh.
Cool.
What do they look like when they're getting ready to release eggs?
There was a bunch of little white dots that were in its tissue that are like the little eggs that, if you've seen like the videos of when corals seed and they release all of those.
So even though they're all different critters, they all release eggs at the same time?
No, so it can vary. But they actually can track it pretty well. Like in shallow water corals, they can time it out. So scientists will all go to a research station like the week that they think these corals are going to release their eggs, and go diving at night to try and catch it, because it happens at night. You go there with your flashlights, and
it only happens at night.
And they can sometimes time it down to the day.
Oh wow!
Wow.
Yeah.
But like only at night?
Yeah.
Regardless of being at 2,000 meters depth?
Well, this is at shallow
Okay.
locations, right? So here's a bit different.
So, but
full wide.
all of the polyps
Coming up.
on like one of these deep corals would all release eggs at the same time?
I'm not sure, Sam, are the deep corals the same?
We, for the most part, do not know. That's one thing I actually love about deep sea biology is there's so many very basic questions left to answer. There's a few, I think there's a few examples of brooders and a few that we think broadcast spawn, but it's not really clear if they are maybe just locking into like a tidal signal to coordinate that, or yeah.
Yeah, and that's one big difference between these corals and the shallow water corals is people are able to spend, you know, all day diving or snorkeling on these corals, looking at them, observing them, understanding their patterns, whereas, you know, we're down here for 20 hours and this is the first time people are really seeing these areas and these corals, so it's really hard to get those predictions.
Right.
Yep.
So a brooder would be like, that's like a chicken, right? Sitting on its eggs?
Sure, yeah, there's more kind of investment into each larvae versus broadcast spawners, like you're just making 1 million and hoping that 3 of them survive to adulthood.
Okay, I might be getting a little into the weeds on this, but how does that change? Like, do corals that are brooders, like, is it obvious? Like, does that change their morphology much? If they're investing a lot of energy into their baby corals? Or is it really
I'm not
hard to
aware
tell?
of any way it would change their morphology. Definitely if they, like, if you can collect any that are currently brooding, you can obviously see that.
Okay.
And corals can reproduce both sexually and asexually, right? Is that true?
Yeah, so we were talking about this a little bit last night. So for like stony corals, which we haven't really seen on this cruise yet, fragmentation is like, I wouldn't say like a primary reproductive mode, but it's definitely like one of their reproductive modes. So pieces will break off and just like keep growing. So you have what you can really, consider to be a new colony, even though it's clonal, right? So it's the exact same genetic material. So a lot of the stony coral reefs that we see, it's kind of this interesting mixture of a lot of them are clones. Like some reefs, I think I've seen like 30% might be clones. So there's not quite as much genetic variation.
Cool.
Does the thing that happened with few genetic variation on mammals, for instance, happened also on other species? Like when you have few genetic variation that you may have problems and blah blah blah. Does that happen also to corals, for instance?
I'm not sure what you mean.
Like lack of genetic diversity will start putting in defects.
And making it susceptible to disease.
Yeah.
Oh, yeah, sure. I think in general, a genetically diverse population is a little bit more robust. Just looking, for example, at like ocean acidification or warming, if you have a degree of warming and you have a lot of genetic variation, maybe there's some subset of the population that has a few genes that make it a little bit more resilient to warming.
Okay.
And then those would be the ones that survive and reproduce more.
Yeah.
And if you don't have that standing variation in the first place, then yeah, that population could be in more trouble.
Okay, yeah, makes sense. We're seeing a lot of this type of fish today.
Boring fish.
Yeah. I think it's the only one we've seen, right?
No, we've seen another one at least one other.
Okay.
Which one was it, Albert?
I don't know, but it has the angry face.
The angry face one? Okay.
Albert, I would buy your book, man, I'm telling you. Biology and Geology of Doug Seamount.
Are we still on Doug?
No,
we're not on Doug anymore.
That's a name, Seamount. That's Baker Island, actually.
But I think we did just officially name Doug Seamount.
It seems to be sticking.
We came up with no other name, so.
And that one has a lot of charisma, really.
To be fair, Albert did have that beautiful name that was like a connection to his village, and it reminded him, and then we were all just like, but Doug.
But the name of my village is saved for my village.
Yeah, that's true.
Oh, that's true.
It already exists.
Albert, how far away are we from that web marker there?
Web marker there.
Yeah.
1,100 feet, which I don't know how far that is because I don't do feet. So, feet, 360 meters.
Okay. Yeah, it's funny. For most things, I do think in feet, but then with ROV dives, I can only think in meters because that's all I've ever done.
Yeah, at one point in my life, I was almost capable of thinking in feet and inches and all these really nice measurements that you English people use. Well, English speaker people use. That was when I was living in Scotland for 6 months. But at the moment I step foot back home, that was gone.
Zoom in there. What's that?
A black shrimpy?
Yeah, did you see the other day I saw? I think that's
almost
a black
like
shrimp.
purple.
Yeah.
Is that just me?
It
a
does
white?
look
No, it looks
purple to
purpley.
me, yeah, like a dark purple. It's cool.
That's
So
a
you
good
see,
shot.
we will have to agree with Sarah at the end. There are things interesting in the sand.
Yeah, I know. I'm telling you.
All right, full wide.
Coming out.
Could you only imagine trying to coordinate so many legs?
I know. I have a hard time with wrist too.
There's another one.
Bridge nav, step again, please. Thank you.
I think if I remember correctly, things like centipedes don't really coordinate the legs in a sense. Like each leg has its own little neural cluster.
Oh, really?
That runs it. Yeah, and it's basically just like a pretty simple algorithm of like the leg next to you just did this, so you're gonna do this.
Yeah, but at some point you need coordination, otherwise you are not going to go towards where you want.
Well, I think there's probably a more central neural system that's saying
couple
go
of
there,
neurons.
but you know, it's kind of like when you're running, but you don't really think about what each leg is doing, right?
Yeah, well, yeah. But
Or
that
that's
doesn't
how
mean
I
that
run,
they
I
are
don't
not
know.
coordinated. They are coordinated, no?
No, it's coordinated, but it's coordinated in some ways by a few very simple rules, by like little clusters of neurons in the legs themselves.
Oh, what's that?
Ooh, looks like an urchin up there.
Blob.
And a fish.
And a fish.
Ooh, this frame has so much biology on it.
Hey, zoom in there.
Yeah. Yeah, that's the kind of thing that I hit with my feet on the beach the other day. Yeah.
Oh,
Yeah,
that's
that's
really pretty
painful.
though.
Yeah.
Wow.
It's cool. You can see the kind of like the 5 bars coming out of it, so it kind of looks like a star when you're zoomed in.
Yeah.
Oh, yeah.
See?
So is that a pattern that happens to all of them, or?
Well, it's an echinoderm, so it's related to sea stars.
Ah, so wait, that's a sea star.
It's a sea urchin, but it's related to sea stars.
Like a cousin.
Same phylum.
Yep.
What's that thing? It has like white pointy, like white ends on the.
All right, full
On
wide.
some of the.
Coming
Are
out.
those? I think they're like venom sacs. I think is what the other dive was saying. If I have it right, that some of those
Bridge
the
nav,
spines
step
have
again,
little,
please.
almost like poison sacs on there.
Poison harpoon dart things
Yeah.
too.
Yeah.
So pretty friendly.
Yeah. Real snuggly.
Well, they lure you in because they're so pretty. And then, bam.
Yeah, you just want to snuggle it, but no. Not advisable.
Not advisable at all.
Yeah. Now Argus is pretty far behind. Probably the next step I will wait for it to swing slightly back, because otherwise.
Yeah, it's actually fairly steep here. Just soft.
Oh.
Some rock
You've
stuff
been
coming
feeling
up.
like you were falling behind, but Mike, it was really because I was looking forward.
Yeah, no, yeah.
Okay.
I'm just watching in a down camera.
Okay.
coming up.
Yeah. Well, according to the bathymetry and the backscatter, we should be getting closer to harder substrate. There you go, a rock.
Rocks, yay.
Oh, there's a coral there, it looks like.
Yeah, so it's a large primnoid on the, actually can we zoom on that? Looks like probably Caligorgia.
Go ahead, video.
There's also one on the bottom left, well, two.
Yeah, we have 3 corals all of a sudden, this is great.
Wow.
Yeah, so large
We
primnoid.
found them.
We found them.
Yeah, if we could do zooms on all 3 of these, that would be great.
Yep.
What part do you want to see most?
Video?
This has been great, and yeah, once we get that kind of, yeah, we can do like polyp zooms. Perfect.
I'll go down and look at the bottom and then come up.
Albert, we're pretty
Yeah,
laid
not-
back. Okay.
Yeah,
Yeah, he's just saying we're gonna stop and check that.
yeah. You are like 20 meters behind your actual position. We can step back if you want for like.
Yeah, if we're in a good spot, I'd like to spend just like a minute here.
We may be okay, because Argus is going to sort of float over the top of Herc and settle out. Maybe a little far away, but we have some time.
It's incredible seeing these brittle stars just stretch out on the coral. It looks so fragile, but it's holding a lot of weight in brittle stars.
Yeah, totally. It's amazing just one leg can wrap
Hey
so
Sam,
many
if you
times.
guys are in position, you could sample this one.
You want this
Okay,
this
we'll
Caligorgia
probably
or
need to rush
Caligorgia
it
like
back
one
a little.
that we're looking at right now?
Yeah.
Okay. Yeah, if we can get in a good spot, we're gonna actually try to sample this.
That's going to be 240, and that's going to be like 20 meters.
Okay.
Bridge nav,
Here,
can
come
we
wide
do
for a minute,
20
video.
meters at
Full
240,
wide?
please?
Yep.
20
Coming out.
meters at 240, please. Thank you, Vitaly.
Hey, go in on that pen there.
So that's a bamboo coral. Seeing a bunch of these on this cruise.
So is there a kind of rule of thumb on how far apart the rings are on the bamboo corals, or they can be closer or farther away depending on the species or the...
Yeah, you know, I'm not sure, Albert. That's a good question. Bamboo corals definitely come in a lot of different morphologies, even in what we think are either very closely related stuff or even the same species. So, yeah, I'm not sure about the little nodes, though.
All right, pull out a little bit.
Is that okay? You want more?
That's fine. Just go back in on the yellow guy.
Do you have your altimeter on Argus?
It's as far in as I can go.
It's generally been turned
Oh, that's
off.
great. Thanks,
I think it's
Nick.
off now.
What's up?
No, we are getting close to something, and I wanted to see your altimeter.
Okay. Yep.
Yeah, pick
Bridge
Argus up.
nav.
a bit.
Cool, so
Argus, we need to
Make
probably
another
come
step,
up with
please.
Yeah, I'll come up
Herc.
with you. Full wide video.
Coming out.
Just get ourselves sorted here.
Yeah, so I think we're going to try and sample that big white one whenever you're in a good
Okay.
Yeah, just bear
position.
with us for a minute.
Oh, yeah,
and
take your
manage
time.
Argus here.
Yeah.
Yep.
We were cruising at high speed.
Yep.
So
And the annoying scientist had to ask for something.
Yeah, no, it's fine. We just need to settle things first.
Yep.
Okay, I've got Argus at a...
I put another step back.
Okay, that's great. Argus is at a reasonable distance with
Yeah,
a 33 meter
that's not gonna
delta.
work, though.
Oh,
You're
yeah,
gonna
yeah.
have to come down.
It'll swing back.
Yeah, now the ship has to come still like 15 meters that direction. So
yep,
that should put us in a good position for working on that spot.
The big fan. So this is a Caligorgia, which it looks like it is to me. It's one of these taxa that really closely related species look like almost identical. And so you really have to look at it under like electron microscopes and look at the sclerite morphology, or you can tease it apart just with depth zonation. So if you know, okay, we really only have this species at this depth.
Yeah.
Or obviously you can just barcode the DNA.
Do you do in the lab? Do you do like you have like water pressure chambers for studying deep sea corals, or how do you do that?
So a rule of thumb, and I honestly have no idea who originally came up with this rule of thumb, but rule of thumb is that if it comes from like 1,000 meters or less,
no longer being tucked.
you normally just don't have to worry about
I
that.
think you're going to want to come back so I can get the scope. Right now you're upslope.
Yeah, I'm still waiting for Argus to come back.
So I'm going to need like 25 meter delta when I'm down there.
Yeah, for sure. I'll come down as soon as that slope moves away from me. Argus has still got like 20 or 30 meters to catch up with the ship, so that'll make it safe.
Ship's now almost in position, so.
Okay, yeah.
Eventually
And
it will
Argus
swing
is still.
back. And there's no current apparently, so it should swing back straight down aft.
Can you tell me why the depth on ENAV or NAVG, whatever, is not accurate or not logging at all?
Because we are extremely depth.
Too deep for the software?
No, I don't know. Yeah, it's this and the heading of Argus, it's not properly displayed. These are the two only things that...
Oh, that heading of Argus might be off as well?
Yeah, that's...
Yeah, no,
that's
it not might be off. It is off.
It is off. Huh. Well, John Hallen's coming out in a few days.
Right.
Give him a list.